Thank you, Min, for the kind introduction, and good morning to everybody. Like Min introduced, I actually work with platelets and platelet extracellular vesicles, and that is also my disclosure for this talk, which is titled Origin and Physiology of Blood EVs. So please come and discuss if you disagree with what I've been saying afterwards. So it's quite possible that you don't agree, because I'm not an expert on erythrocytes and leukocyte EVs. Okay. So this is how most of us will think about the blood. We just focus on this part, and it's either plasma or serum that we often get our hands off. And as Rink introduced very nicely, so I don't have to go into it, it's good that you know what is the difference between the two. And we will be discussing it today, but there's also the beginner's guide that will help you to have that basic understanding. So that's important. But blood is much more complex than that. It is the dynamic and liquid matrix, the plasma, and then there are different cells that are contributing to the complexity. And Rink also mentioned already the different centrifugal fields, what can happen depending on the time and the G-forces that you use. Blood is viscous, so it's possible to separate the cells and the plasma into different fractions, as shown in this schematic image. And under certain centrifugal fields, it's possible to get about 50% cells, erythrocytes, in the bottom, and on the top, about half of the liquid as in plasma. And in the middle, we have the so-called buffy coat that contains the leukocytes, with variable cell contributions, and the platelets. And it's very important to remember that, these cells really outnumber the other populations. So that if we think of the leukocytes, which are rich in variety, they are minor in the numbers, and they are outnumbered by 50 to 40 times by platelets and 100 to 1,000 times by erythrocytes. So if we would think that all the cells in blood would contribute to the EV, blood cell EV population in the circulation, you would think that we would have a vast number of erythrocyte EVs, but we are beginning to understand that that might not be the case. Then what I want to show next is the clinical blood smear, because this is very much getting more into the physiology. So the red things here are the erythrocytes, and you can nicely see how they outnumber the tiny, tiny blue dots, for example, here and here, which are the platelets. And then you can see two large leukocytes, most likely neutrophils. And this gives you a very good idea about the distribution. And clinical blood smear is a very good tool for doctors to spot abnormal morphologies and abnormal cells in the blood. I'll come back to that later. So if we want to move forward to more realistic understanding about the blood, this image is already getting there. Why? Because it has the plasma shown very nicely. The cells are more in proportion, and we include the endothelium. So blood is not in a tube, it is actually in the vessels that are surrounded by the endothelial cells, as already mentioned also by Rink. And the endothelial cell type, they depend on the type of vessel they are in. So whether it is arteria, arterioles, vena, or venules, or the capillaries, the endothelial are all different. And the properties of the endothelium are depending also on the shear forces that they encounter. So the blood flow generates shear forces, and for instance in arteria it presses to the side the smaller particles. As far as I know, it hasn't been shown for EVs, but most likely in the arterioles also the EVs get crushed by the endothelial side, and it's therefore easy to understand that they might be promoting to the endothelial permeability and the fitness. And the last slide here is just to show that the blood vessels cover the whole body. So we are working with a very large organ system, even though it's in the liquid form. And you can nicely see that the major organs or the tissues that we think that are also contributing to the blood EV population are very rich in in vessels. So this just a sort of like short Physiology 101 to keep us reminding what we are talking about and the complexity. Okay, enough talked about lipoproteins already, but just to mention again that the lipoproteins really outnumber the vesicles and the cells in the circulation, all 6 magnitudes higher, and that the properties of the lipoproteins also overlap those with the vesicles, making isolation and quantification a challenge. I would more like to talk about the plasma proteins because that is much more ignored so far in the complexity discussion. So many of us want to do, for example, SEM. This is SEM unrelated to EV research. It's just an artificial matrix. But if you have wanted ever to take pictures of blood cells in SEM or EVs, you easily find out that you get this nice coverage of plasma proteins and you don't see anything. It's also a confounding factor in your Western blots. So this is what we are having to do with. So, but if we just think of plasma proteins, and I already mentioned that plasma is viscous and very rich in the proteins, there's 60 to 80 milligrams per milliliter of those proteins, we are forgetting something very important. Firstly, what I want to point out is that it's only very few types of proteins that constitute the majority of the plasma proteins. Importantly, the albumins and the globulins, including the IgGs, I'll come back to this later, and fibrinogen. And fibrinogen is the source material for the fibrin network, which is very important to stop the blood flow in the case if we get a cut, or in case of infection when we need to restrict the access to blood to certain places. So these have very important roles in the blood. But there are over 1,500 different protein species in plasma. So they may be a rich source for biomarker materials. And many of us are interested in the blood or the plasma or the serum just for this sake, not because of the blood EV physiology like myself. So for a long time, our goal has been to obtain as pure vesicles as possible. But in reality, the vesicles are covered with molecular corona, protein corona as called before. And what we are learning to understand more and more is that it can be, and this comes from the nanoparticle studies from pharmaceutical side, or nanoparticle studies in blood, that the corona can be divided into hard part and the soft part. And what this means is that how accessible those proteins are for exchange in different environments. What is emerging in the field, and I'm not showing any data or quoting any papers here, that this corona may impact the physiology of and the roles of what the blood EVs are playing, for instance, influencing the uptake and what the EVs can be doing. But I want to give you an example of biomarker research, and this is a very nice paper from Hoshino et al. In Cell 22, where they compared over 400 human samples and looking for cancer biomarkers. And what they found out that there was a very good predictive power for some immunoglobulin subunits, marked here in orange, and that they really could discern the cancer patient samples from the human samples. And in this study, Torres et al. Compared 3 different methodologies to purify the EVs, very common UC and SEC, and then combining SEC with UC. And you can see that these 2 immunoglobulin IgG subunits were lost when you were isolating with SEC and UC. So you might be losing also some of your biomarker capacity if you go for very pure EVs. This is just food for thought. Okay, so what are the cellular sources of EVs in circulation? So I already said that we should think about the ratios of the blood cells. That might be an impacting factor. And many of us are interested in the very, very small proportions of EVs that might come from tissues or, for example, from cancer. And we are getting very good advancement in the single particle analysis technologies, such as flow cytometry. But as Rink showed already, that we can't see the smallest particles and we can't necessarily see the largest particles. It might be due to how we process the samples, but the whole spectrum is difficult to get in any one technique. So there have been few studies that have gone about trying to understand the contribution of different cells to blood EVs in a different way, and this is by analyzing the content. There is a study that is analyzing the RNA, and then there is analysis of the proteins. And I just really shortly explain what this is about. So in this paper, the researchers isolated the EVs and then the RNA content, and then they used very fancy bioinformatic technologies and analysis to deconvolute the data, and therefore to detect the source origin of the cells that were contributing to the EV populations. And what they saw was that about half of the blood cells were EVs were coming from platelets, and surprisingly B cells, if we think about the proportions of the cells in the circulation. For tissue origin, they found that the majority of the EVs were coming from adipose tissue, but we had also contribution from smaller contributions from other tissues. Now, this may depend on the isolation of the EVs, because also there's another paper that I'm not showing here in Nature Biotechnology by Volberian et al. From 22, and they just isolated cell-free RNA, and they got very different contributions for the cellular RNA. So it is good to keep in mind that the results are very much depending on the material and the analysis methods. What about the proteins then? Well, in this nice, very recent study, they were looking at the serum proteins and comparing the serum protein catalog with those of tissue-specific EV proteins. And they could show that there was presence of proteins from all these tissues in blood. So good for us. So this would mean that if we want to have biomarkers, for example, for liver, we can detect them in blood. Well, then what influences the amount of the EVs in the blood? So I think there is this homeostatic status. So there is a certain amount of biogenesis, and then there is uptake and clearance, and all is good. And then it can be perturbed under certain conditions. And how the EVs, for example, from tissues are entering the blood, it's a very good question. So most likely they are picked up by the lymphatic system from the interstitium. There's 8 to 12 liters daily in a person processed, so that the lymph is contacting the blood. But also under disease conditions, this homeostatic balance can be disrupted, and there are more EVs forming, and they could have access to blood when the endothelial barrier is breached. And this image is from this paper that has nothing to do with EVs again, but nicely showing how the tight contacts between the endothelial cells become loose under certain disease conditions. So, in case of pathologies, we can have more EVs entering the blood from certain tissues, but also in a healthy physiological situation, there are conditions where we can have more contribution from tissue EVs in circulation. And my cases are from muscle and heart during exercise, and then from a placenta during pregnancy. And let's take the pregnancy first. So the placental EVs are thought to contribute to the tolerance to the fetus in the maternal, promote the maternal tolerance towards the fetus. Sorry, I said it the wrong way around. And exercise EVs are very interesting. It's been shown that there are more EVs in plasma from muscle during exercise, and there are now already in vivo studies in rats and mice showing that those plasma EVs can contribute to cardioprotection. And there are even studies that show that there's some cancer mitigation if we compare to mice that have been treated with plasma EVs from sedentary rats. So the cancer studies in rats. So it's worthwhile thinking about that there is this capacity in the plasma EVs, not knowing where it's coming from, but most likely something to do with the muscle, because the comparison is between exercising and sedentary animals. So then for the beginners, I'd like to, as a beginner and trying to go through with all what has been published with the physiological roles of blood EVs, it is a nightmare. So to give a small framework that you could start off with, I've chosen to divide the physiological roles of blood EVs thus. So divided into roles played by removal of material, roles where intercellular messaging is involved, and then an active role for the EVs, an active role for the surface. And why the latter? Because the plasma EV volume of the total volume of the EVs per mil of plasma is very small, but actually the surface is very large. And if we would take into account the high turnover that we will soon hear in this meeting, we can think that the surface role is much more important than we have considered so far. So some examples for the removal. So transferrin receptor in exosomes. Everybody knows this because that's how exosomes were first described. Also cells that are facing apoptosis can maintain healthy status by removing caspase-3. Or if they are under attack from complement, they can remove the complement material in EVs. Cancer cells certainly want to get away with the chemotherapeutics, and then there are processes that we don't yet understand very well. What are the molecules that are getting disposed of, but there are things happening in cellular aging. And this can work also as a decoy functions. What we are very interested in is the roles that are played between cells. What I mean with this is that there is certain cargo in the EVs, and then when a recipient cell takes it up, it changes either its function or its own cargo, such as transcriptome. And there are so many different examples in the literature for what blood EVs can do. So this can be related to the microRNA in EVs, the cytokines on EVs, antigens or the organelles, and I come back to the organelles at the very end of my talk, viruses and so forth. And these roles are played in very important physiological functions such as immune functions, wound healing, aging, and so forth. So this is just a large category. And finally, there's sort of active roles. So I consider that there are two independent roles for blood EVs, and one is the maintaining of the hemostasis, so coagulation. And but the surface is actually capable of supporting also anticoagulant properties or fibrinolytic properties. So it's all a matter of context or the environment where this vesicular surface is being provided. But that's sort of independent, but it needs to be initiated. And at the normal condition in blood, there is no coagulation, because that would be, of course, counterproductive. And another independent role, I think, is the antigen presentation, and I have some examples when we get to the leukocyte roles. And finally, we don't have a very good idea about the molecular corona yet, but hopefully we soon will understand that role in physiology as well, soon. So in the next few slides, I will give you some examples for the physiological roles for the blood cells, or the EVs from the blood cells, starting with the erythrocytes. And this is very unfamiliar terrain for me, and I feel uncomfortable to talk about this when, for example, Min is in the room. So, but I already mentioned that there is the exosome formation with the removal of transferrin. Also, EV formation is very important for the erythrocytes when they age. You need to remove material and make the cell membrane soon such that the old erythrocytes can be removed. What I found very interesting in this review was that what they said is that the EV formation is not correlating with the hemolysis. So we have liberation of heme and hemoglobin, but not EV production. I would like to hear whether that is true, but I'm just telling you what the review said. And what I found interesting is that it's been shown that erythrocytes can form two populations, this large and small EVs, and they seem to have different capacities to handle oxidized proteins and promote the endothelial or protect the endothelial permeability, as mentioned in the beginning. What about the leukocytes? So there are many different flavors in different proportions, and this is like really vast amount of literature because there's been a lot of research ongoing. And I would kindly refer you to this very excellent review by Professor Bujas from 22. And I'm highlighting a few things. So first one is the many different types of EVs and biogenetic routes that can be taking place specifically in leukocytes. So if you can't properly see the text, but if you go to this review, you can see that there are many different ways that are different from the usual microvesicle exosome routes. And to me, this suggests that this is very important for the physiology. Then in this slide, I summarized all the leukocyte EV roles. So I've divided them into three. So adaptive immunity, antigen presentation as an independent function. And in addition to just the usual how the cells will learn to recognize foreign things in the body, this antigen presentation is very important physiologically when the lymphocytes are being developed. So how the T cells learn to recognize self from non-self, and also for B cells. And there is EV formation every time there is an immunological synapse, and that must have a strong physiological role. Then there is the delivery of all kinds of interesting immunoregulatory molecules and cytokines. I put them as a separate. Here is a list of some of the molecules that have been found. And cytokines are interesting in that respect that they can be inside the EVs, they can be on the EVs, or they can be soluble. And it has been shown that there can be synergistic effects physiologically relevant if the cytokines are provided both in the EVs and from the soluble fraction. And finally, the really big bowl of inflammation and innate immunity. And the only thing what I'm saying about that is that it's, again, a balance. So we are always talking about the homeostasis, and it is the microenvironment that regulates which way things will go. And then to my favorite topic, only 2 slides about that. So first, the blood smear to show abnormally that megakaryocyte is in the blood. And this is to remind you that the source for platelet marker material can be both megakaryocytes or platelets, and the jury is still out on what is actually happening in blood circulation. And then about the platelet EVs. So they can be formed under many different physiological or pathophysiological conditions. And also they have interesting different biogenetic routes, suggesting that this EV formation is very tunable and probably very regulated depending on the need of the body. And this is highly dependent on the different receptors on the platelet surface. So platelets are like sentinels in the circulation. They can take up cues via receptors, get activated, generate signaling, and then generate different EVs. And this is something that we have been working for a long time. So I want to show you data that is submitted where we activated the platelet through different pathways, isolated the EVs. We had 4 different populations, and these are all through receptor activation, and then we compared them to EVs that were formed from non-stimulated platelets. And then we wanted to see if they are different and how they are different. And the nutshell result is that we couldn't really discern the differences with the traditional physicochemical characterization or the omics studies, but we do see vast number of differences when we give them to macrophages and look at their secretomes. So we need functional assays to understand the physiology of different EVs. And this was just to show that we put them in zebrafish and we could get them to macrophages. So I also would want us to move to more in vivo studies. And then the very final thing, what I want to say, I promised you I will talk about the organelles, is the delivery of mitochondria. And this, of course, you know the size of mitochondria is not happening in the small EVs. But it has been shown that, for instance, platelets deliver mitochondria that will improve neutrophil and macrophage mitochondrial fitness. And this really interesting recent study in Nature Aging this summer shows that plasma EVs, small plasma EVs given from young mice to old mice will improve their mitochondrial energy metabolism. So maybe George Lucas was onto something with the midichlorians. May the force be with us, and thank you.